the paper tests this hypothesis, therefore, that you actually proposed back in your initial submission of your telomere work to Nature back around the turn of the millennium.
2000.
Yeah. That infections increase, past infections increase rate of frailty in later age. And so, there's a lot more to be said, but maybe you want to pick it up from there.
Yeah. What I wanted to do, when I saw this paper, I was very excited because, as longtime viewers and listeners will know, the way I work, I'm a theoretical evolutionary biologist. And so the coin of the realm in theoretical work is hypotheses that make predictions that allow you to later test them. And when they are tested by you, or better, by somebody else who doesn't have the perverse incentive of having provided the hypothesis, when the prediction turns out to be true, what it does is suggest that the model that was used to generate it has some credibility. And so, as longtime viewers will know, I did some work starting in somewhere 1998, working through 1999, on an evolutionary hypothesis for the pattern that we call senescence. Senescence is what, in common parlance, we would call aging. There's a reason not to use the term aging. Aging is imprecise, but senescence is the thing that causes the body to grow feeble and inefficient with age. And I came up with a model that united what we knew about the evolution of senescence with emerging work in molecular and cellular biology. And in any case, that work has produced a number of predictions which over the intervening years have turned out to be true, reflecting positively on the model. To be very brief about the model is that we as organisms, I mean, this is true for most vertebrates, presumably, and certainly all mammals, we are composed of hundreds of billions or trillions of cells. In the case of an adult human being, you probably have 30 trillion cells at any one time as a full-size adult, and most of those cells have the potential to become tumors and then cancers, which are obviously potentially fatal. So in an organism with that many cells, in which most of those cells have the potential to turn lethal and kill you just simply by becoming deaf to the signals that they shouldn't grow anymore, evolution has built a system that reins in tumors that begin to grow or would be tumors, what in the paper that Debbie Ciszek and I ultimately published, we called prototumors. So prototumors are things that are one step short of becoming a tumor, and the one step short makes them a patch of cells that has grown out of control and then self-arrested. And one of the predictions of this model was that all moles and probably freckles are manifestations of cells that have been damaged because they're on the skin, probably damaged by UV light, that have run away from their developmental program and begun to grow in an unregulated fashion, and then been stopped at something called the Hayflick limit, which was known before we did our work. The Hayflick limit arrests cells at a number of cell divisions that is programmed in general, from birth.
Which is different for different cell types.
Yes, it is. And
the Hayflick limit is a number for any given cell, which is different between cell types and presumably between cell types, within cell types between organisms too.
Oh, yes, absolutely. And it is set in development differentially, so that our argument in the paper was that selection will set all of those different limits so that they provide you the ability to replace that tissue for a full-length life. And any excess capacity you have gives you a cancer risk that isn't compensated for. So selection has an incentive to back off the limit, to reduce your risk of cancer, and to provide you enough repair capacity to get through your life. If you do a kind of work that damages one tissue over and over again, then, if you're a guy who operates a jackhammer, your elbows are probably going to age faster than other people's because you're doing more damage. Every day you're damaging.
Repetitive stress injuries, same thing with, damaging your lungs repeatedly through inhaling combusted compounds.
Yeah, if you're a smoker or a coal miner, you're accelerating the aging of your lungs. If you're a heavy drinker, you're accelerating the age of your liver. Liver, a tissue in which you have a anomalously large capacity for repair. Why? Because your ancestors encountered lots of things that damaged livers, and so having
And its job is detox.
right. So anyway, we ultimately published in 2002 this model, which we tried to publish in 2000 and were turned down by Nature. So Jen is showing the 2002 paper that was ultimately published. Anyway, there's a long, sordid story about the blocking of publication of this work and how it ultimately made it into publication. But the interesting thing for me in reading this paper,
The Ragusa et al. Paper that just came out.
is that it says there is a relationship between infection and senescence, the degradation of the body.
Yeah, and I was thinking actually their 40+ point frailty index is basically a quantitative measure of senescence.
Yep.
Yep.
Now, when I saw that, I was very excited because that was a prediction.
But not, you just said relationship. They have attempted to describe a causal relationship in which earlier infection predicts later frailty.
Yes.
Earlier, and I believe that they have done so.
Yes, and I believe the work is very good and that it demonstrates this. Now, interestingly, they didn't spot our work. It doesn't surprise me so much because our work was not as widely circulated as I think it should have been. Yep. So they may have been unaware of it. They also didn't mention telomeres or Hayflick limits. So I found that an interesting omission. It doesn't degrade what they've done, but it does sort of reduce its power for no reason. Now, they do mention it obliquely. You want to read the sentence in the paper?
Me?
Yeah.
Yeah. Let's see. So you can show my screen if you can show my screen. So this, again, is the paper we were showing earlier, Can Infections Drive Frailty? Insights from the Baltimore Longitudinal Study of Aging. That just means that the data that they used were data that had been collected previously, and then they went in and tested hypotheses with those data. There's a number of things that I've highlighted in here, but there's a particular thing here. We're still in the introduction. And so this is not their work. They are setting up the problem that they are investigating. Line 77, 78, infections could lead to frailty through mechanisms including heightened inflammation, tissue damage caused by pathogens, or faster cellular aging due to increased cell turnover. And I will say, I know you're going somewhere, but I went and looked up their reference 10 that they've got here. And it's this, that didn't help. It's this paper called Aging and Infection by Gavazzi and Krauss. I was trying to find people who work with Adobe know that this happened. So this is published in Lancet Infectious Disease the same year and early in the year that your paper was published. And so they also don't cite you, but I think their paper actually came out before you guys. And this is a very well cited short sort of theoretical review of the idea, of the very idea that is being postulated in the new Ragusa paper.
All right. So I want to just read a section. So what I have here, I actually, for reasons that aren't worth going into, I didn't have access to the electronic version because it's on a different computer. I actually dug out the physical file that contains our original submission to Nature. It contains the recommendation letters from George Williams and Dick Alexander to Nature that they review it and publish it. So it got the widest possible circulation. It's got their rejection letter with the absurd claim here.
I think it's, let me see if I remember. This is not of sufficient general interest.
It said, we do not feel that it would be of sufficiently strong interest to a general audience to justify publication in Nature. Now, this is a paper that unites, it builds a model that addresses cancers and senescence, builds them together, and hybridizes them with the well-established evolutionary theory of senescence penned by George Williams, that came to them with a recommendation from George Williams and a recommendation from Dick Alexander. So George Williams and Dick Alexander are both members of the National Academy strongly recommending to Nature that they take this paper seriously. This is from Dick Alexander's letter. He says, I wish to offer my strongest support for the manuscript herewith submitted by my 2 doctoral students, Bret Weinstein and Debbie Ciszek, titled Life's Slow Fuse, Telomeres, Tumors, and the Evolution of Vertebrate Senescence. This is the first written support by me for a student paper in my 43 years of academic life, during which period I have guided more than 30 doctoral students in evolutionary biology at the University of Michigan. Anyway, he goes on. But the point is, very strange. This is 2 members of the National Academy, which is the elite of the elite, are recommending that Nature take this seriously. It is about cancer and aging, 2 very important processes that have very important medical implications. And Nature says they do not believe that the paper would be of sufficient interest to a general audience to be worthy of publication.
It was like a pull down menu of reasons to reject.
Right.
And they just picked one.
Yep. It's what's called a desk reject. They didn't even send it to a reviewer. The editor just decided, not worthy of our attention. We're too good for that. So anyway, the fact that the paper keeps producing,
keeps revealing that it had accurate and risky predictions in it.
Right. Things that are manifest, including, as people who have heard this story before know, the prediction that the long-established ultra-long telomeres of mice that suggest that telomeres themselves are not an important regulator of senescence in mammals, because if they were, a tiny short-lived animal wouldn't have extremely long telomeres. This invalidated that work through a collaboration with Carol Greider, who, working with her graduate student Mike Heeman, tested my hypothesis that wild mice wouldn't have long telomeres, and therefore established that the breeding colonies were producing long telomeres, which have a direct implication for drug safety testing, because what long telomeres do is give mice short lives that end with cancer and give them an indefinitely large capacity to repair damaged tissue. So if you give a toxin to a mouse, you may actually extend its life because you're giving it effectively a form of chemotherapy that is harder on its cancers than it is on the rest of its body.
If you give a toxin to a lab-bred mouse using these protocols which breed them early and kill them young.
Right. And then if you give that same toxin to a person, you will shorten their lives. And if you were to give it to a wild mouse, it would shorten their lives. But because we're using these broken mice without knowing that that's what we are doing. So anyway, there's all kinds of interesting stuff, including that highly unusual prediction. Why do I say highly unusual? Because how often does evolutionary biology, working strictly from logic and the evidence in the literature, successfully produce something in a cellular molecular biology lab of medical importance? It's not a common phenomenon. So anyway, major success that the model was able to predict at that level. But here's another one. Yes. You now have a paper in 2026 strongly suggesting a causal link between infection and accelerated senescence through frailty. So I want to read to you a section from the paper that Nature refused to publish because it wasn't.
So this is your original, or
this is the original?
This is not published in Experimental Gerontology, or is
no? This is the submission to Nature, never published anywhere. It was revised into that paper, published in Experimental Gerontology. It was shorter. We had to cut things out in order to get it in. But what we actually thought, what we were ready to publish, is all here in this original longer manuscript. So in a section called "One Source, Three Sinks." And I'll try to translate this, the part that is unavoidably technical. Vertebrates use reserve capacity in growth, maintenance, and repair. Reserve capacity being how much replacement of tissues you can do in a lifetime. And each process, that is to say, growth, maintenance, and repair, erodes telomeres, reducing proliferative potential, reducing the amount of later repair and maintenance that you can do. Through antagonistic pleiotropy... Though antagonistic pleiotropy and accumulated damage hypotheses have traditionally been viewed as alternative explanations for senescence, the finite reserve capacity approach, the model we present in this paper, integrates them.
Do you want to explain antagonistic pleiotropy and accumulated damage, or does it not?
Yeah, no, it's probably a good idea, as long as we're here. Okay. You are composed of something like 30 trillion cells. Your genome has something like 20,000 active genes. On average, those active genes have something like 5 different edits apiece. So at maximum, you have something like 100,000 genes to work with. When you compare 100,000 genes to 30 trillion cells of 200 at least different cell types, you don't have a lot of genes in order to result in the remarkable complexity of the organism that is produced. What that suggests is that the genes in your genome don't all do one thing. In fact, almost all of them do multiple things. We call that pleiotropy, when one genetic element has multiple consequences. Because of the fact that your genes do multiple things, it will logically be true, and this is George Williams' work, which was written in 1957. George Williams argued that your genes will do multiple jobs, they're all multitasking, and that some of those jobs will give you a benefit early in life. They will give you what he called youthful vigor at the cost of inevitable senescence. So his point is there's a trade-off. And my dissertation was on trade-offs. I was inspired by Williams and the power of what he had done in this case. And I said, what if trade-offs is a way to look at all of biology and understand all the things we don't yet understand? And wow, did that turn out to be powerful. But his point was there will inevitably be things in your genome that benefit you early at a cost late. When that happens, selection will tend to favor them, even if the late life cost is high. Why? Because the earlier stuff in life counts more, evolutionarily speaking, than the late life stuff. If you have a pathology that strikes you dead at 80, you've done most of your reproducing, you've even done most of the passing on of your wisdom to your descendants. So the point is the cost to you of dying at 80 from something is comparatively low. In fact, lots of people never make it to 80. So you have a pathology that kicks in around 80. You get away with it scot-free if you get run over by a bus at 60 or eaten by a tiger at 37. So the point is, it's always a bargain to pick the stuff that helps you early in life, even when the delayed cost is substantial. And so Williams, building on Peter Medawar's work, where he argued the force of selection drops off the older you are, Williams said there are trade-offs, and when there are trade-offs that benefit you early at a late life cost, selection will collect them, and our genome should be full of these antagonistic pleiotropies. Pleiotropies, that is, genes that do two things, working at cross purposes to themselves. He said the genome should be full of them, and that accounts for why the body degrades over the course of a lifetime. A very elegant theory. It was a theory at the point that I got to the work. Mm-hmm. Why was it a theory? Because indirectly, we could test many predictions of that model in nature, and we could see, for example, that possums that lived on islands where the predators were reduced tended to live longer. They aged slower because their likelihood of living longer was greater, and therefore the cost of those late life effects was greater to them. So selection backed off the system. So anyway, we had many of these tests where in nature we could see that the exact strange patterns that George Williams had predicted turned out to be true. We knew his theory was at least very close to right, but we hadn't found any antagonistic pleiotropies. So anyway, that's where I entered the picture.
And you say in the bit from the paragraph that you were reading from that your and Debbie's hypothesis here integrates antagonistic pleiotropy, which you just described, with the other main model, which is accumulated damage, which is just exactly what it sounds like. It's very simple. You know, the idea that you just accumulate damage over time, and therefore at some point you're going to hit some limit in some tissue that sends it into a tailspin and you're gone.
Right. So neither of the models that were pre-existing were fully satisfying, either in the antagonistic pleiotropy case, because we didn't have any genes that matched the description George Williams said should be common in the genome, and accumulated damage didn't work because the body is capable of producing new cells. So in what way? It's not like you're a car that wears out. You're like a car that comes with a factory that can produce a new alternator every time you need one. So why is the car getting worse? So this fixes that and says, actually, these are two sides of the same coin, and that there is a limit to cellular reproduction The reason for the limit is to prevent you from getting cancers, which would overrun your body before you ever got around to reproducing if you didn't have this limit.
Again, that limit was already established by Hayflick.
By Hayflick, yep. And it was increasingly understood that telomeres were the likely thing that set the Hayflick limit. And as you point out, the Hayflick limit is set on a tissue-by-tissue basis in development, and our argument was based on pre-existing evolutionary theory that it should be set in a way that the body kind of comes apart in a coordinated fashion over time rather than everybody dies from, the failure of their liver because it doesn't have a high enough limit. Okay, so though antagonistic pleiotropy and accumulated damage hypotheses have traditionally been viewed as alternative explanations for senescence, the finite reserve capacity approach integrates them. Damage, even if it is functionally repaired, will accelerate the aging of tissue by limiting the capacity for future maintenance and repair.
There's mechanism right there.
The liver of a heavy drinker, for instance, may function essentially as well at 40 as it did at 25, but should fail more rapidly than the liver of a non-drinker, even if alcohol consumption ends before damage is evident. Any factor that damages tissue, including mutagens, pathogens, mechanical wear or trauma, oxidative stress, and free radicals, will promote the local increase in a tissue's rate of senescence. So
Beautiful.
That's the prediction right there. Pathogens are on the list of things we said would borrow from the ability to do future maintenance and cause you to age faster. And here we have an empirical...
More frail.
Yeah, and become more frail. And here we have an empirical result that says that seems to be true and causally linked. Slam dunk. All right, feel good about that.
Beautiful, as you should. It's fantastic.
Now, as long as I was here, I wanted to point out that actually on the prior page of this is another one of my favorites from this paper that I believe has turned out to be highly valid based on later work.
Oh, is this going to be my favorite too?
It is going to be. Now, I do have one regret here, which is, although the name that Debbie and I gave to the process that we were describing here is a very good name in its own right, it confused people and therefore didn't really even have a chance of catching on, I think. The term is histological entropy. And the problem with the term histological entropy is that many people reading a paper on senescence saw the word entropy and they thought we were alluding to a long discredited explanation for senescence, which is that the body, like everything else, degrades from reasons of simple...
It's accumulated damage.
Yeah, well, it's not even accumulated damage. It's just basically thermodynamics.
Oh, yes. Okay, different.
Right.
Yes.
So to the extent that people read the word entropy and their eyes glazed over, they didn't take in the meaning. But
yeah, but so you're going to read from that. But I have, since you first proposed that, just thought that it captures perfectly what you are describing. And even at that time, we were in our late 20s, early 30s, at a moment in life when most people, if viewers are at that age, you already have begun to see that tissues in your body and repair does not happen as smoothly and seamlessly and invisibly as it did when you were 18, right? You heal from injury, you heal from a wound, soft tissue, tears so smoothly when you are 18 that it seems to leave no trace. But as you age, the repair itself of tissue, histology, histological entropy, becomes more scattershot.
It becomes more scattershot for a reason that, let's put it this way, this idea has been reinvented, and it's been reinvented under the term epigenetic drift. Now,
I don't like it nearly as much.
Epigenetic drift, I believe, is the same idea, and what it means, and what you will hear we proposed in this paper, is that there is an informational component to senescence. If you think about the way you came into being, you started out as a zygote. Sperm meets egg, produces a single cell. It's got all of your genes present. And then that cell goes through a programmed series of divisions. And as it does that, it becomes, each of those cell divisions, the first two cells look very much alike. And the farther you get down that road to being your 30 trillion cell adult organism, the more narrow each of those pathways becomes. So at some point, your arm develops and the cells in your arm don't have to worry about being a liver or an eye or anything anymore.
They're no longer totipotent. They're no longer stem cells.
Right. Totipotent means they have the potential to become anything, like your zygote or early stem cells. Pluripotent means that you have the ability to become many things, and then you become committed to a particular function over time. And so you can imagine in the isolation of the womb or the egg or however it happened to you, in your case, it would have been a womb, but in that isolation, it's a very controlled environment.
You don't know how many chickens are watching. Sorry.
That's true. And I wouldn't want to count the ones that are watching and have not yet been hatched for reasons I'm not clear
sorry,
on.
that's so derailing, but
turnabout is fair play on this podcast. But okay, so you're in this isolated environment. The maternal body protects you from all sorts of outside influences. And so you have this elegant environment in which these cells can go through all of these doublings and the cells can become increasingly committed, and they know exactly where they are, right? The cells are in a position to know how far down the road they are from their initial zygote single cell stage. And therefore, they can produce an organism in a very predictable way because there's a certain number of cell divisions you have to go before you start producing an eye. And then there's a certain number of cell divisions you have to go before you start producing, a retinal cell.
Yeah. So they have a knowledge of number of divisions, and they have a knowledge of position and neighbors.
Right. And so that knowledge results in a well-formed baby, assuming you have a good genome, which you almost certainly do, and assuming that it has been insulated from insult like chemistry, which increasingly you can't guarantee because the mother can only filter out so much stuff, and the novel stuff often gets through because she doesn't have any built-in mechanism to deal with it. And don't vaccinate a pregnant person. Right?
That should be obvious.
You would have thought. Mm-hmm. But in any case, the cells know a lot about what they're supposed to do next when they are properly insulated from the outside world and the genome is well structured, which it almost certainly is, or it wouldn't have gotten to this stage in the first place. But if we then fast forward and you take that person at 30 years of age and they damage some piece of tissue, right? They, get a burn. Well, now you've lost a bunch of cells. And the ideal thing to do would just be to regrow what was there from the outside. But you can't, because for one thing, the cells that put themselves in the location that you've now burned are not from the same starting position as the cells that will now be filling in. What's more, there's been damage over the course of those 30 years, and it has continually eliminated cells that knew exactly where they were and replaced them with cells that only approximately know where they are. And the more approximations, it's like a Xerox of a Xerox of a Xerox. Yeah. For those of you old enough to remember Xeroxing. But in any case, the point is there's a loss of information from the system. So it is informational entropy that Debbie and I were pointing to in this paper. Mm-hmm. And let me read that to you now. So this section is titled Somatic Senescence Due to Cellular Attrition, that is loss of cells, and Increasing Histological Entropy, loss of information. To our knowledge, increasing histological entropy is the scrambling of cells based on the loss of information. That's what I mean to say, to be precise. To our knowledge, no explicit mechanism linking Hayflick limits to the phenomenon of vertebrate aging has been proposed. We offer the following approximation. Development continually increases histological differentiation and specialization, that is, cells becoming more and more narrowly defined, which are maximal when an organism becomes a reproductively capable adult. That is also the moment at which senescence begins, interestingly. It doesn't begin when you're born. It begins at reproductive maturity. Throughout life, damage and programmed cellular turnover result in cells being lost from the soma, that is, the majority of your body, not your gonads, and being replaced. When cellular lineages exhaust their reserve capacity and are lost, they must be replaced by neighboring lineages if they are replaced at all. We propose that the uncompensated loss of some cellular lineages, coupled with the replacement of other lineages by neighbors adapted to slightly different roles, diminishes the optimal arrangement of cell types. By our model, body-wide senescence results from the combined effect of A, uncompensated cellular attrition, that is the loss of cells, and B, increases in what might be called histological entropy, both of which will diminish an organism's efficiency at accomplishing whatever tasks differentiation initially evolved to address. Senescence of this type should progress at a non-linear rate, accelerating with age as fewer cellular lineages maintain and repair an ever larger proportion of the body. And then we go on to provide examples. The aging of human skin appears to progress as our model predicts. Skin thickness decreases by 25%. That's the uncompensated cellular attrition between the 4th and 8th decade of life, and entropy increases. And we then quote from another paper. The epidermis of an older individual exhibits a marked variation in thickness, often in the same histologic section, and a disparity in size, shape, and staining quality of the basal cell nuclei under a light microscope. There is also a loss of the orderly alignment of cells along the basement membrane and a disruption of the gradual upward uniform differentiation present in the epidermis of younger individuals. Electron microscopy studies show that the basal cells of the flattened dermis of old individuals lack villi. Deletion and derangement of small blood vessels is found in aged skin, with sun-damaged skin being most severely affected. So that is a place where damage from the sun has resulted in the acceleration of the informational loss and the derangement of the tissues. Anyway, there's more to it, and I will also say later in this paper, someplace I have not highlighted here, we connect that idea to the issue of the mice with their unusually long telomeres as a result of a defect in their breeding colony. Because what turns out to be true is that what is true of human or other animal tissues when you look at them under a microscope, which is that they become increasingly chaotic and deranged the older the individual you got them from is not true of the mice. The young tissue looks like the old tissue. Why? Because they have an indefinitely large capacity for replacing their tissues. Voila. So, all right. Anyway, I thought it was cool another prediction showed up, and I would like to set a rule.
I think this is a rule you've tried to set before.
You think so?
Well, I don't know the rule you're going to say, but what's in my head, I think you're going to...
We'll see.
Let's see.
I will probably for the rest of my life be trying to set this rule. The rule is this. A lot of people think I'm stupid, and they may be right. However, I don't want to hear that I'm stupid from anybody who hasn't produced something like this at least once in their life. I am perfectly willing to entertain the hypothesis that I'm an idiot from anybody who has, but everybody else should shut up on the topic.